All right. So, hello everyone. Welcome back. So today we'll be reading the methods section. They should be going over how they taught undergraduate students how to peer review articles. So let's jump right into it. Methods, context, intervention, and participants. In a research liberal arts college for female, transgender, and non-binary students in Mount Holyoke College, we implemented a curriculum on peer review in two different contexts. A, as a full 14-week seminar course, Course 1, Peer Review in Biology, or B, as a single unit of peer review activities embedded within a disciplinary biology course, Course 2, Vaccines. Both courses were offered as upper-level electives and taught by the same instructor, Arcel, in the same semester, spring of 2022. In Course 1, with 9 students, peer review activities were scaffolded to transition the student from a novice to an expert through 4 units, based on the clinician training paradigm of see one, do one, teach one, and the gradual release of responsibility model of literacy education that uses the framing of I do, we do, and you do. Okay, so that was a whole bunch of information just right there. So it looks like their audience or their study group were female, transgender, and non-binary students, and that is their test group. And they had 2 different styles of teaching. So they had a full seminar course and a single unit activity. And it was called Course 2 Vaccines. Okay, so generally upper level electives are for graduate students, or not necessarily graduate students, but they are either for graduate students or high level undergraduates. So undergraduates are also allowed to take upper level electives. I'm thinking of so it's either high level undergraduates such as juniors or seniors. There we go. Okay, so course 1 had 9 students. And they were scaffolded. Basically means the lessons build upon each other. So lessons build upon each other, and then they transition from novice to expert through 4 different units. All right. Based on the clinician training paradigm of see one, do one, teach one. I see. So basically you see how it's done, you try it yourself, and then you teach someone else. And that is, I guess, how clinicians are trained. And the gradual release of responsibility. I do, we do, you do. So basically, when you're in training, especially when I was in training in research lab, first my supervisor showed me how he did it, and then he taught me how to do it, and then finally I did it by myself. So that's I do, we do, you do. Okay. Figure 2 provides a conceptual overview of the full curriculum taught in Course 1. In Course 2, with 11 students, only one minimal unit, i.e. do one, where students perform peer reviews, was implemented to complement a discipline-specific course on vaccine biology. In both classes, the instructor and or students selected biology preprints of interest to review, and then critically analyzed the preprints in writing and in discussion using guiding questions provided by the instructor, and then wrote peer review reports as a professional peer reviewer would do. See context, peer review activities below for further details. Okay, so in Course 2 had 11 students with only one minimal unit. So I guess there was only one do one unit was implemented to complement a discipline-specific course on vaccine biology. Oh, so they specifically selected a rough draft or a manuscript to peer review that was related to vaccines. And in both classes, the instructor and our students selected biology preprints of interest to review. So they got to pick their own research articles. And then they analyzed them, and they discussed it using guiding questions. Okay, students submitted weekly reflection journals in response to prompts about their perceptions of their performance, sense of self-efficacy, and understanding of disciplinary literacy in the context of peer review. A majority of students indicated their intention to publish their reviews publicly online to document their expertise and participate in the professional COP. Community of preprints, I'm guessing. I'm not exactly sure of that one. Students also interacted with preprint authors and other experts in peer review through email and video interviews. All students provided informed consent to participate in the study, which was verified by the Mount Holyoke Institutional Review Board as exempt, according to 45 CFR 46 101B, Educational Research and Test Surveys, interviews on October 12, 2021. So as any good research study does, generally you have to get consent from your test subject so that they know what they're being involved in. You don't want to do test experiments on unsuspecting, poor unsuspecting like people on the street. Okay. Context, curriculum, and peer review activities. So course 1, peer review and biology students engaged in 4 peer review events throughout the full curriculum. Okay, so there were 4 peer review events. What those events are, we have yet to find out. Okay, so event 1, review 1, individual review on a manuscript selected by the instructor. The initial baseline event was assigned as individual homework after the first class meeting. After an initial discussion of the concept of peer review, the initial baseline event was assigned as individual homework after the first class meeting, but after an initial discussion of the concept of peer review but before students had carried out any in-depth training in the course. So I guess if we're to draw out a timeline here, this is like a timeline. It was assigned as individual homework. So first class, get homework on peer review and preprints. And just a reminder, preprints are basically rough drafts that have been uploaded publicly online. After an initial discussion, so they told them what peer review and preprints were, but they hadn't had any in-depth training yet. So no training. Basically, it's like a pre-test. Okay, all students reviewed the same manuscript selected by the instructor for its accessibility to a general biology audience. So basically, they picked an easy one that college students could understand. The manuscript was written by pre-college students, submitted to the Journal of Emerging Investigators, and ultimately published as 31. Okay, so that should be a reference at the very end of the document. Event 2, Review 2, individual review on a preprint selected by student groups. The next event took place in Unit 3 after about 6 weeks of explicit teaching about peer review and after students had co-created a rubric to evaluate preprint peer reviews. So they created a rubric that was, I'm guessing, alongside the instructor. And they also had 6 weeks of explicit teaching. Students were grouped by interest in biological topics, i.e. cancer and COVID-19, and then each group selected one preprint to review, which was approved by the instructor as being accessible. Each group member individually carried out peer review of the same preprint as homework. Students were not provided with detailed written instructions for how to write a peer review, Instead, they were asked to create the review based on the learning from Units 1 and 2. Guiding questions for peer review were provided as an optional resource, but answers were not required to be provided in the assignment. Okay, so not provided with detailed written instructions. Basically, they got to write how they wanted. Okay, event 3, group review on a preprint selected by student groups. Students shared their individual review too with their group members, then spent time in class discussing their individual reviews and the instructor's feedback on them. Then they synthesized their individual reviews into one group review. After completing the group review, students reread their individual reviews and self-graded using the rubric they co-created. Okay. So they synthesized their individual reviews into one group review, and they also self-graded their review using the rubric. Okay. Event 4, Review 3. Individual review on a preprint selected by individual students. The fourth review event was assigned as an individual final assessment after the completion of the curriculum. Without any input from the instructor, each student selected a preprint of interest and wrote an individual review. Okay. So, without any input from the instructor. And then they wrote an individual review. So basically, they're doing it all on their own. Course 2. Vaccine students engaged in a single module of the curriculum and 3 peer review events. Event 1, Review 1, individual review on a manuscript selected by the instructor. The initial baseline event was assigned as individual homework after a 30-minute discussion of the concept of peer review and after 6 weeks of disciplinary lessons on vaccinology. All students reviewed the same preprint selected by the instructor for its accessibility to a general vaccinology audience. I feel like a general vaccinology audience is still not very general, but because this course did not involve explicit lessons on peer review in class, students were instead provided with detailed written instructions for how to perform a peer review, including guiding questions which are required to be answered as part of the assignment. So they got more, this group that only had one lesson were given more instructions. So basically they were told to go do it themselves. Okay, event 2, review 2. Individual review on a preprint selected by student groups. Students were grouped by interest in vaccine topics, and then each group selected one preprint to review, which was approved by the instructor as being accessible. Each group member individually carried out peer review of the same preprint as homework, using the same detailed instructions and guiding questions as review 1. Okay, so they all did peer review of the same preprint or rough draft as homework. Okay, group review 2. Group review on a preprint selected by student groups. Students shared their individual review too with their group members and then spent time in class discussing their individual reviews and the instructor's feedback on them. Then they synthesized their individual reviews into one group review. So similar to the curriculum 1, they synthesized their individual reviews into one group review. And then they discussed their reviews and the instructor's feedback. Okay. Assessment of disciplinary literacy, peer review quality. So this is the big test because the big deal with not teaching undergraduates peer review and how to give feedback on research is that currently there's a belief that undergraduates are basically too stupid to give good feedback. So if this is successful, hopefully, these undergraduates should be able to provide feedback that is at the same level of quality as an expert who has lots of knowledge in biology. So let's see how this goes. Assessment of disciplinary literacy, peer review quality. All peer reviews written by students were de-identified by the instructor and provided to the independent researchers to assess within student development of peer review ability longitudinally. The researchers generated 4 metrics of peer review ability using 3 unique tools. Ooh, okay, so this is going to get into the more technical aspects. So, to assess within student development of peer review ability longitudinally. Basically, that means you're assessing the same students' peer review ability over time. That's what longitudinally means. I really wish they just used simpler language. This makes it hard to analyze myself. All right. This is glitching. Oh well. The Review Quality Instrument, RQI. So this is they created a new measurement to help measure the quality of peer review, specifically for this experiment. So RQI consists of 8 Likert scale questions ranging 1 to 5. One question asks the evaluator's overall opinion of the review, and this question is reported here as RQI. The other 7 questions ask about the components of the review, and here these have been combined and reported as RQI total. Okay, so I'm not an expert in necessarily qualitative tests myself, so I don't know what the Likert scale is. But generally, it's not really too important to know all the details of a methods section, as long as you get the general idea of how they are measuring their tests. Because unless you're an expert in these fields and want to redo the experiment yourself, that's when the specific tests become more important. But generally for number 1, I would say they are basically just on a scale of 1 to 5, how would you rate the quality of this review? That's what I'm guessing the question was basically. So these evaluators were just answering this question. And then after they did this, asked 7 other questions, they would total it all up and you would get a score from 7 to 35. Okay, the second measurement they used to gauge the quality of preprints was the pre-review review assessment rubric. So pre-review is actually another organization that specifically focuses on reviewing preprints. So this is actually an organization. Okay. So a rubric for evaluation of preprint reviews was generated by the students in Course 1. This consists of a series of nonlinear scores of 0 to 4 being awarded to different sections of the review, which are then converted into a percentage and reported here as MHC. So nonlinear, I kind of like this word and I kind of don't like this word, but basically anything that is not a line or is not basic multiplication or addition when you're calculating a score is considered nonlinear. So maybe you might be doing like percentages or fractions and you are like calculating them in different ways. That's not a very good explanation. Basically, as long as you're not multiplying or adding scores together, if it's not simple math, then it's probably not linear. So so basically, the important part here is that Another measurement they used to measure the quality of the preprints that these students, the peer review that these students did, was their own rubric. So use the students' own rubric to judge the quality of their peer review / feedback. So additionally, scientific literacy more broadly was assessed using Gormley's TOSLS survey administered before and after each of the 2 interventions. TOSL's results are presented in Appendix 1, and appendices are generally sections at either the end of a research article or in a separate PDF document that just have extra data. So, thematic analysis. Students completed weekly reflection journals, which were de-identified by the instructor, provided to the independent qualitative researchers, and assigned pseudonyms to retain anonymity. So de-identified reflection journal entries were uploaded to MaxQDA and coded by thematic analysis. Thematic analysis used students' science literacy, science identity, and belonging within the scientific community to inform initial latent codes. I'm guessing this is how initial latent codes means the base like for a test it would be used as like the base value that you compare everything to. There we go. Okay. Initial themes corresponding to science literacy included understanding science content, using science skills to help others, talking about science with others, and practicing science now and in the future. That says science a lot. So these codes were then collapsed into the following overarching themes: knowledge, understanding and talking about science; practice, applying performing science skills and knowledge; and value of practice, understanding the use and need of science. Okay, so saying these initial latent codes are basically the initial themes that people most often wrote about. We also identified personal and environmental variables corresponding to a student's identity that were then organized as professional identity. So pursuing a science career or internship and interacting with science professionals. And personal identity. So systematic / structural barriers and access to resources. So this is one thing that is kind of a pet peeve of mine in science, where basically there's very obviously like ethnicity gaps and other obvious gaps in science. So like a lot of people at the research lab I'm in are either Caucasian or Asian, or or like not necessarily Indian, but they call themselves brown. I don't know if that's offensive, but unfortunately there aren't that many black students that are in research. And so that's why you'll see a lot of initiatives nowadays trying to get minorities and opportunities for minorities to participate in research as research assistants or other opportunities. But that was my little tangent. Anyways, back to the research article. So finally, we divided codes or themes relating to belonging into presence, absence, or facultative, i.e. a sense of belonging in some contexts but not others. It's a weird word. I've never heard of the word facultative before this. Anyways, to identify differences between each course context, we compared codes across courses to so basically to compare the differences between the two courses. Compare the differences between the two courses. So that's what this one is. And to establish trustworthiness, two team members separately coded 20% of the students' reflections and ensured a minimum 90% inter-rater reliability. So basically, two of the research team members who were doing this experiment they went through the students' reflections and they would identify their reflection as like presence or maybe professional identity or basically all of the other themes that they set out. So that's what coding means. They just like read the article and say, this article, this section of the student's reflection is about this theme. And then they made sure that the different analyses between the researchers were 90% similar, at least in 90% similar. So we also engaged in expert debriefing to discuss the alignment between our themes and our theoretical framing. So they mentioned earlier that their theoretical framing was how they wanted their thematic hopes, I guess, or the hopes that they wanted to accomplish with this experiment. So that was like more scientific relatability and feeling more included in the scientific community. So those overarching themes that they had for this experiment. Okay. Inclusion and exclusion criteria. Only data from students who completed all assignments were included in the analysis. So this represents all 9 students in course 1, and then course 1 was the multi-week course, and 10 out of 11 students in course 2, and course 2 was the vaccine-specific workshop. In course 2, 1 student did not complete reflection journal entries, and so that student's data was excluded from the thematic analysis. Another student did not complete the peer review exercises on time, nor the post-survey for TOSLs, and so this student's data was also excluded from those analyses. Okay, so then the next section is for the results. So that's where the actual goodies are. But in general, the point of the methods section is just to prove to a reader that their experiment was makes sense, so that they're not just pulling numbers out of their butts. So it's basically to prove how they got their results. And if they have a good experimental design, so if they design their experiment well, then their methods should make sense, and so then you can trust, or you can more likely trust any results they get. Overall, though, I wished they went a little bit more in depth into the actual curriculum in their methods section. They might have that in the appendices at the bottom. I have not checked that yet. But why I say that because I would love to implement this curriculum myself, because I want to learn how to review preprints since I'm also an undergraduate student. So, since we are done with the methods section, I will be doing the results section on Tuesday. So oh dear. If you want to know how these students did, then come back on Tuesday. But if you have any questions for today, leave them in the comments below. I will try to answer them as best as I can. And also please like and subscribe the video. I will see you all on Tuesday. Bye!